Hey, welcome to The Explainer. Today we are diving deep into one of the absolute cornerstones of biology, DNA translation. This is it, the process that turns the genetic code in your cells into the actual proteins that make you, well, you. And look, if you're prepping for an exam, you're in the right place, because we are going to walk through every single detail you need to know to ace it. All right, let's jump right in.
So here's our game plan. We're going to start with the big picture, the basic definition. Then we'll crack the code, the genetic language itself. From there, we'll meet the molecular machinery, the tools and the factory, and then we'll watch the three-stage assembly line in action. We'll even cover the finishing touches, and finally, we'll see how all this knowledge plays a huge role in modern medicine.
All right, first things first. Before we get into all the nitty-gritty details, let's make sure we're all on the same page. What exactly are we talking about when we say translation?
Okay, so think of it this way. Translation is basically the cell's construction phase. It takes a temporary blueprint, a molecule called messenger RNA, or just mRNA, and it reads that blueprint to build something real, a chain of amino acids. And that chain, called a polypeptide, is what folds up to become a working protein. It's the magic step where information becomes function. Right, so now we know what it is. The next logical question is how? How does the cell actually read the blueprint? To answer that, we have to learn the language. Let's decode the genetic code. Let's start with a number, 64. That's it. That's the total number of words in the entire genetic language. See, the cell reads the 4 letters in RNA, A, U, G, and C, in groups of 3. These 3-letter words are called codons. And when you look at all the possible combinations, you get exactly 64 codons. That's the entire vocabulary for building every protein in your body. Now, these 5 characteristics are super important, especially for exams. The code is unambiguous, meaning a codon like GGU will always mean the amino acid glycine, no confusion. But it's also degenerate or redundant. This means multiple different codons can code for the same amino acid. That's a fantastic built-in safety net, right? A random mutation might not even change the protein. The code is also nearly universal for all life on Earth, it's read continuously without punctuation, and it's non-overlapping, which just ensures a clean, accurate reading every single time. Okay, this table here, this is your bread and butter for any biology exam. These codons are non-negotiable. You've got one start signal, AUG. It does 2 jobs. It tells the machinery, start here, and it brings in the first amino acid, methionine. Then you have 3 stop signals, UAA, UAG, and UGA. They're like the period at the end of a sentence. They don't code for an amino acid, they just say, okay, we're done.
So we understand the language. Now it's time to meet the molecular construction crew. Let's take a look at the machinery and the key players that actually get this job done. At the heart of it all is the ribosome. You can think of it as a mobile protein factory. It's actually made of 2 separate pieces, a small subunit and a large one. The small subunit is the reader. Its job is to grab onto the mRNA blueprint and find the starting line. Once it does, the large subunit comes in. It's the builder, the part that actually forges the bonds between the amino acids. And inside this factory are 3 critical workstations, or sites. You gotta remember these, A, P, and E. The A site is for arrival. That's the docking bay for the next amino acid. The P site is for processing, where the growing protein chain is actually held. And the E site is for exit, which is where the used-up delivery molecule gets ejected. APE, arrival, processing, exit. You're going to see questions on this for sure. It's the core of how the ribosome works. Now, if the ribosome is the factory, the transfer RNAs, or tRNAs, are the specialized delivery drivers. Each tRNA is a master of one thing. It has a 3-letter sequence called an anticodon that perfectly matches an mRNA codon. This lets it read the blueprint and bring in the one and only one specific amino acid that's being requested. Total precision. So you might be wondering, how does the tRNA driver get loaded with the right package? Well, that's the job of this incredibly important enzyme, aminoacyl tRNA synthetase. Think of it as the tool master or the loading dock supervisor. It makes sure that each specific tRNA gets attached to its one correct amino acid. This step, called charging, is absolutely critical for accuracy. A mistake here means the wrong part gets put into the final product.
All right, we've got the blueprint, we've got the factory, and we have the workers with the right parts. It's showtime. The whole process of translation breaks down beautifully into 3 main stages: initiation, elongation, and termination. Let's watch the assembly line in action.
Initiation is all about the setup, getting everything in the right place to start. First, some Hopper proteins literally grab the small ribosomal subunit and bring it to the mRNA. The subunit then slides along the mRNA, scanning until it finds that AUG start codon. Bam! Once it finds it, the very first tRNA carrying methionine locks on. That's the signal for the large ribosomal subunit to clamp down, completing the factory. And here's a key detail. That first tRNA starts in the P site, leaving the A site open and ready for business.
Elongation is the main event, the repetitive cycle where the protein is actually built. It's like a three-step dance. 1. A new tRNA arrives in the open A site. 2. The ribosome forges a peptide bond, connecting the amino acid chain from the P site over to the new amino acid in the A site. 3. The whole ribosome moves one codon down the mRNA. This is called translocation. The empty tRNA is now in the E site and gets kicked out. The tRNA holding the growing protein is now in the P site, and the A site is wide open for the next one. And this just repeats over and over and over again.
So, how does this all stop? Eventually, the ribosome hits one of those 3 stop codons in its A site. Now, there's no tRNA that matches a stop codon. Instead, a special protein called a release factor fits right in. And this release factor is like a wrench in the gears. It triggers a reaction that cuts the finished protein chain loose from the last tRNA. The new protein floats away, and the whole factory, the ribosome, the mRNA, everything just falls apart, ready to be reused for the next job.
Now, you might think we're done, but hold on, not quite. That long string of amino acids that just got released, it's not a functional protein yet. It needs a few finishing touches, what we call post-translational modifications.
There are 2 main kinds of modifications here. First, and most importantly, the protein has to fold into its unique, precise 3D shape. It's like protein origami, and its function depends entirely on this shape. Often, helper proteins called chaperones make sure it folds correctly. Second, the cell can add or remove chemical tags. Things like phosphates or sugar groups can be attached, and these act like on-off switches for the protein's activity, or even like address labels that tell it where to go in the cell. So why do we need to know every single one of these intricate steps? Because when you understand a process this fundamental, you can learn how to control it. And that is a massively powerful tool in modern medicine. Let's see how. The core idea here is brilliantly simple. If a cell can't make the proteins it needs to survive, it dies. And this is exactly how many of our best antibiotics work. It turns out that bacterial ribosomes are just different enough from our own human ribosomes. This allows us to design drugs that gum up the works of the bacterial factory, stopping their protein production cold while leaving our own cells totally unharmed. It is a fantastic example of targeted molecular medicine. And that really brings us to the future, doesn't it? We've mastered targeting bacterial translation to fight infections. But as our understanding gets even more sophisticated, what's next? Could we develop new drugs that shut down viral protein production? Or maybe even find ways to selectively turn off translation in cancer cells? The power to control this fundamental process of life's construction, well, that's still one of the most exciting frontiers in all of medicine.